大家好, I'm Ying Xu, a professor in Modern Animal Research Center, Nanjing University.
大家好, I'm Zhiwei, a graduate student in Xu Lab.
Now, what we are going to tell you is that we find a mice carry a missense mutation in a phosphorylation site in core clock components period 1, mistime feeding, and predisposed to obesity. As all of you probably realize, we're living in an environment that's cycling. The Earth's rotation around its axis generates a 24-hour cycle of light and darkness. And throughout evolution, many different live organisms have evolved clock systems that predict environment day and night changes, and therefore to prepare for such changes. Clock mutations affect significantly on many physiological processes and behavior. In mammals, there are 3 PER proteins, PER1, PER2, and PER3. Individuals carry out S662G mutation in human PER2 called family advanced sleep phase syndrome. In these individuals, they will wake up about 3 to 4 hours early and go to sleep about 3 to 4 hours early on average than normal individuals. This position is the first of 5 serine space 3 amino acids apart, and the S662G mutation impedes sequential phosphorylation. This motif is highly conserved in mammary PER proteins. Zhiwei, if we mutant this site in PER1 in the mice, maybe we can observe very interesting phenotype.
That's a great idea. Let's try.
Using microinjection, we successfully generated PER1 S714G mutant mice and analyzed the phenotype of this mutant mice. We were first surprised to find that only the PER2 mutant had a third sleep-wake cycle, while the sleep activity rhythm in PER1 mutant looked normal. But I thought sleep is not the only output of the clock, so I measured other physiological rhythms such as time to eat or metabolic rhythm. Look here, mice carrying this mutation showed advanced feeding phase. The peak of food intake showed obviously advanced in mutant mice.
When we first saw the metabolic rhythm in these mice, it was impressive. While PER2 mutants that had an abnormal sleep-wake cycle, they had a normal metabolic rhythm. Conversely, the PER1 mutants that had a normal sleep-wake cycle exhibit very abnormal metabolic rhythm, as if they will wake up in the middle of their sleep to gorge on food. There is emerging evidence that abnormal eating time along with energy deficit diet predisposes to obesity.
Under normal control, there are no obvious differences between body weight among different genotype mice. However, under high-fat diet, mice carrying S714G mutation gained body weight quickly, and this phenotype can be abolished by night-only feeding.
Please look at the microdata. We found the pattern of expression of in these genes was inverted.
Oh, really? Oh, yeah, very striking changes, such as female PER gamma delta clock. Oh, good. So let us talk with sandwich pattern.
These mice had a telltale sign that they were eating very differently. They would wake up in the middle of their sleep, eat something, and then go back to sleep, and kind of maintain that almost every day. Then we had a different idea. We thought, well, we have heard this thing called night eating syndrome. It runs in many people where people actually wake up in the middle of the night and they binge eat. And these people have very much more difficulty in weight control and other stuff.
Indeed, the need to rest, eat, and adjust to daily changes in the physical environment may have resulted in a co-evolution and integration of the circadian clock, metabolism, and the rest activity cycle, particularly in high organisms. Human PER2 S662G mice have been suggested as an advanced sleep phase syndrome model and exhibit a 4-hour phase advance of activity rhythms in light-dark cycles. In this study, we found that human PER1 S714G mice exhibit a markedly advanced phase of feeding behavior and predisposed to obesity.
Now we can uncouple sleep-wake cycle from feeding-fasting cycle by simple mutation in a gene. So that means these two processes are very different, and that will lead us to find what are the neural bases, what are the appetite bases, what are the metabolic bases of these cycles. Second is, even if there is a genetic predisposition to eat differently than a normal animal or individual, if we make this simple lifestyle intervention or lifestyle changes, we can override that genetic predisposition and make an animal, or maybe one day make a person healthy.